Okay, let me begin. So thanks again for joining today and for tuning in on our webinar about protein and DNA sequencing. For those of you that don't know me, my name is Monique Seymour, and I'm a scientific sales executive at Repertoire. So what that means is I work with a variety of scientists in a variety of therapeutic and diagnostic areas. And I work with them to achieve their research goals, either by trying to solve some of their protein and antibody problems, or by providing them with tools to further their antibody discovery research. So just some background on Rapid we are a bioinformatics and mass spectrometry technology and services company located in Canada. We specialize in proteomics and more specifically, next generation protein sequencing, which I will break down for you a little bit later. We are experts in our fields and we've sequenced over 3,000 monoclonal antibodies and we're still going. We've worked with hundreds of companies worldwide. And the reason why we're so good at what we do is because we have our own proprietary technology and software, which we continue to improve. And we also have our amazing specialists that are continuously innovating and are hyper-focused on quality. Now, I had a look through our attendees today, and there are a wide variety of listeners from different scientific backgrounds, including students. So I will try to make sure that I address some of the basics before I get into some of the details without boring our experts that are tuning in. So before we get into the nitty-gritty of DNA versus protein sequencing, I thought I should provide you with a little bit of history on the topic. So as you can see, there have been quite a few developments over the last 70 years. DNA sequencing has, well, it began earlier on, and protein sequencing has really started to kick off probably within the last 30 years or so. However, I would say most advancements happened within the last 15, and that's kind of where Rapid fits in. More recent advancements have really enabled protein sequencing to be integrated in a lot of research processes in ways they were really unable to before, just because it didn't make sense to use. Whereas DNA sequencing, well, everyone is quite familiar with the technology and its plentiful uses. So just a quick market assessment. Clearly, protein sequencing is behind protein sequencing in the size of the industry. But that makes sense as DNA sequencing has been around for quite a while longer, and it is a bit higher on that exponential growth curve. Now, North America is currently the geographical hotspot for both sequencing technologies, holding a majority of the market. But Asia Pacific region is set to grow and take over as one of the largest regions by 2025 and 2026. Now, I am going to take some time to give you a little bit of background on antibodies. And the reason being is that the antibody market is a huge driver of protein sequencing and DNA sequencing. When the antibody market grows, so do the other two. They're very closely linked, mainly because DNA and protein sequencing really enable the research behind antibody discovery and engineering in both therapeutic and diagnostic areas. So the antibody sector is set to grow to value 14.1 billion in 2025. That is a huge number. And again, it is the largest in North America currently, but it is set to be Asia Pacific in the future. I also wanted to take some time to talk a little bit about the restraints and challenges, as well as some of the opportunities and drivers that are going to either contribute or deter the antibody market and the resulting DNA and protein sequencing market. So there is what's called a reproducibility crisis, in which there are some serious quality concerns resulting from a lack of reproducible results, both commercially, academically, and even within the development of therapeutics and IVDs. A study that was completed found that in 2019, global spending on antibodies rose to 3.4 billion, and yet 1.7 billion could have been wasted. And within a different study, 6,000 commercial antibodies were tested, and only half could successfully recognize their specified targets. There's also a severe amount of cost, and the research of antibodies is quite time intensive, which can slow down growth, especially when you may have a lot of troubleshooting that needs to be done to address reproducibility. In regards to methods that could help solve these problems, there is a lot of governmental expenditure globally. COVID has definitely helped with that. And on the other hand, the industry continues to develop technologies to help address these problems related to the cost and the time of development and minimizing reproducibility, so it's no longer labeled as a crisis. As these technologies continue to grow in parallel with this research, hopefully we'll be able to address some of the potential challenges and ideally open up some more areas of antibody research. So to get started on the nitty-gritty science of it all, I'm going to be talking about some antibody discovery technologies and the use of DNA sequencing, as well as protein sequencing to enable it. So we've got Hybridoma technology, next generation DNA sequencing, synthetic library generation, and de novo protein sequencing, also known as next generation protein sequencing. Again, I understand that there are quite a few variations of research backgrounds listening in. So some of you may not be familiar with this content. And if you'd like more information on it, please feel free to send me a message after the webinar. I'm happy to have a chat about it further. So Hybridoma technology, it was created for the first time in 1975, and it was first applied in rats with a generation of rat monoclonal antibodies. Hybridoma cells are generated with a fusion between a short-lived antibody-producing B cell and an immortal myeloma cell. But they can also alternatively rely on immortalization with an oncogene or an oncovirus. Now, each hybridoma cell expresses a large amount of one specific monoclonal antibody, and favorite hybridoma clones are cryopreserved for continuous antibody production over a long period of time. The hybridoma generation process really takes advantage of a host's natural ability to generate functional and highly specific and high affinity monoclonal antibodies. In fact, to date, more than 90% of antibodies approved by the United States FDA are generated by traditional hybridoma technology. So there's no denying its huge contribution to the therapeutics and diagnostics industry. Now, some limitations of hybridoma technology are that the produced antibodies are not always as specific as we'd like them to be. The proteome of hybridoma cells is naturally prone to splicing, mutations, and fusions. So antibodies produced through these means could result in the accumulation of problems that require further analysis through mass spec-based proteomics, as DNA sequencing can provide further insight here. Now, on the image, or in the image on the right-hand side, we see a hybridoma cell that created one or two productive alleles of the targeted antibody gene. The blue and green antibody is the correct IgG antibody, but as you see with everything else in the picture, there are quite a few variations of the antibody alleles, resulting in a lot of not so monoclonal antibodies. In fact, one study showed that 1 in 3 mouse antibodies have more than one productive allele. DNA sequencing is often used to sequence hybridomas, but a limitation is that it can't observe these extra chains, post-translational modifications, or mutations. And problems like this are really what contribute to the reproducibility crisis that I mentioned earlier. Now with next generation DNA sequencing, also known as NGS, there are two slightly different methods. The first is B cell repertoire, or BCR, and the second is single B cell sequencing. Now, both methods result in obtaining the DNA or RNA of the IgG gene. In single B cells, sequencing the peripheral blood mononuclear cells, or the PBMCs, are isolated, and then they're sorted using flow cytometry to sequence individual B lymphocytes. Whereas with BCR, the cells are homogenized prior to sequencing. So one big contrast here is that with single B cell sequencing, because the cells are isolated, you can actually obtain the antibody heavy and light chain pairing, whereas with BCR cells, because they're homogenized, you can't get that information. Now, in both cases, the resulting antibody sequences are recombinantly expressed in mammalian cell lines like CHO or HEC293 to obtain the overall antibody. One limitation with single B cell sequencing relies on culturing individual clones, which could result in a loss of key B cell clones from the BCR. Additionally, novel antibody discovery through NGS does require the spleen to be collected from animals, which could result in the death of the animal. And even though NGS is a high throughput system on its own, it doesn't completely reflect the circulating antibody repertoire, as it can bind to variations in alleles and post-translational modifications due to the fact that it is DNA sequencing. So I personally think that synthetic library generation is a fun one. Here we have B cells that are isolated from plasma to extract mRNA, and then RT-PCR is used to generate a synthetic repertoire. The repertoire is generally displayed in vitro by phage bacteria or yeast. And in the picture we have here, we have some fun little phages. Now these libraries have the ability to produce synthetic antibodies that could actually exceed the affinities and the specificities of natural antibodies. Moreover, these libraries can be recycled to screen against other target proteins. So the display technology is very much dependent on nucleotide sequencing. So again, NGS would be applied here to generate the synthetic library for subsequent affinity maturation. A limitation of nucleotide sequencing approaches, the limitation of nucleotide sequencing approaches can be applied here. So the pairing of the heavy and light chains during the actual library construction could possibly not reflect the natural antibody repertoire. And in fact, light chains or heavy chains could even be missing from the final library. Additionally, because affinity maturation happens in vitro, the antibodies that are generated do not have the relevant post-translational modifications, such as glycans, that would be required for stability. So the initial testing could be blind to the impact that these post-translational modifications could have later on. For example, if there's a glycan on a CDR region of an antibody. The unfortunate result here is that display technology's artificial nature could in fact prolong target validation before an antibody drug could actually be adopted into the clinic. Now, de novo protein sequencing or next generation protein sequencing is a very different approach to antibody discovery. One of the main differences being its protein sequencing, and it involves the use of the natural antibody or serum. It can actually be used to obtain monoclonal antibody sequences from polyclonal serum or to obtain the sequence of a purified monoclonal antibody. It's completed by using multi-enzyme digests, mass spectrometry, and our own proprietary machine learning algorithms, in which the sequences and post-translational modifications are extracted directly from the protein. It really provides a holistic picture of the humoral immune response. And with novo protein sequencing processes, it doesn't require the culling of the producer animal, as it could be done directly on the protein or purified blood sample. To complement efforts in existing pipelines and characterize the broad BCR, de novo protein sequencing can be applied as either proteomics only or proteogenomics approach, depending on which stage of the antibody discovery process it's being implemented. And one limitation with sequencing monoclonal antibodies is that the purity of the sample must be quite high. Now, to build on the information I just provided you with regarding knowing the protein sequence, I'm going to tell you a little story. So we had a client come to us a couple of years back, and this client was one of the big pharma companies. And they came to realize that the protein that they had previously DNA sequenced and expressed wasn't binding like their original one. Now, there are many things that could have gone wrong here, including parts of the expression system But they had checked that, and none of that had ended up being what the problem was. So they came to us, and we helped them out by sequencing their protein. And what we found was, well, through our next generation protein sequencing, was that there was an additional glycosylation that the scientists weren't previously aware of. And that glycosylation was actually interfering with the binding of the antibody. So this is a very clear case in which DNA sequencing alone caused some problems and resulted in some troubleshooting. And they're by no means the only researchers that have come to us with this problem. Now, based off of our experience within the antibody discovery industry and the applications we found ourselves being a part of, we've determined that next generation protein sequencing can actually enhance as well as accelerate antibody-centric research. Now, to be clear, I am by no means saying that it can replace DNA sequencing's contribution to antibody research, but it can absolutely address DNA sequencing's limitations and even complement it as a whole. So if we take a deep dive and looking at the actual code of DNA and amino acids, we can see that one does not directly transfer to the other, even though it's called translation. Despite there being over 20,000 human genes, there is not an equal output of proteins. The 20,000 human genes are translated into millions of proteoforms due to post-translational modifications, alternative splicing, and germline variants, which is why the amino acid sequence detailing the actual protein is so important. Where DNA and RNA have 4 nucleotides as code, proteins possess 20 different amino acids as their building blocks, which could also be modified. The sensitivity of amino acids is undoubtedly higher than that of proteins or of DNA. Think of it this way. Would you be more likely to trust the quality of a study that has had 4 participants or 20 participants? If you're looking to sequence an antibody that's been previously produced, using its cell line, there could be a lot of background noise resulting from degenerated DNA, which will undoubtedly make it more difficult to obtain the sequence of the protein or the antibody in question that binds to your target. Using physiochemistry to characterize your protein is really important because, as we saw previously, there are millions of proteoforms, and these post-translational modifications can impact the activity of the antibody, which again could result in longer target validation times and more time spent troubleshooting. Finally, we have codiversity. Now, this diversity can result from the antibody itself with different alleles, as we saw previously, and it can also result from a range of species. So if you take a look at this picture, we can see that there are not only a variety of antibody isotypes, but if we zoom in on the IgG, there are also a variety of subtypes. And within that, we have a variety of idiotypes and allotypes, all impacting the activity of the antibody. Now, we care about this because DNA sequencing doesn't always address this diversity, but next generation sequencing can. Therapeutics and diagnostics are being developed using a wider variety of species and accessibility to technology capable of finding a protein sequence indiscriminate of species origin is becoming increasingly important. So to provide you with more context on what next generation protein sequencing or NGPS is, I've included a brief outline of the services that we offer before I get into some of the services' actual applications and impacts within the industry. So first off, we have our remab, which provides the full amino acid sequence of a monoclonal antibody. It was our first service offering and what we built off of to create our newer ones like repab. Our repab service is a technology that captures monoclonal antibodies directly from a subject's blood sample, which gives you direct access to the functional native molecule and the ability to produce recombinant antibodies. We also have MatchMab, which doesn't exactly use next generation protein sequencing, but it does use associated proprietary algorithms to analyze a protein and determine that its protein sequence matches the expected sequence. And finally, we have NovaRig, which decodes the immune response at different time points by mining the antibody repertoire in greater depth, along with the combined analysis of B cells and antibody proteins. Now, with all of these solutions, speed, reliability, and quality are at the forefront of the values that we have when doing this work, to ensure that the final products meet the required expectations to enable antibody-centric research. Now, we've worked with a variety of scientists across the biotechnology industry, but specific applications of our services in protein sequencing have fallen really into these 5 applications, which include therapeutics development, diagnostic development, reagent development, protein characterization, and patents and other regulations. Now, I am aware the last 3, reagent development, protein characterization, and patenting, can be thought of as falling under the umbrella of therapeutics and diagnostics, but there are special cases for these areas, which is why I'll be going into detail about each one. So first off, we have therapeutics development. Now, when developing therapeutics, knowledge of the antibody's 3-dimensional structure is critical to guide a rational drug design. Obtaining the de novo protein sequence can really facilitate characterization of the protein itself. And recently, our technology was actually used to help characterize 2 potent hantavirus antiglycoprotein monoclonal antibodies without relying on a cell line or genomics or transcriptomics data. Our technology has also helped enable antibody engineering for humanization. So typically, biopharmaceutical developers would rely on a hybridoma generation or phage display to develop humanized antibodies against antigens of interest. However, homologous monoclonal antibodies originating from other species may already be characterized and commercially available. The sequence of both commercially available or in-house generated anti-animal antibodies can be obtained with 100% accuracy and coverage using Remab. Now, with the knowledge of the primary structure, different versions of the antibody could be generated to suit the research need, whether that be to engineer a chimeric version or to tweak areas to make it a better therapeutic candidate. In regards to antibody fragment engineering or alternate variants, such as bispecific antibodies or diabodies, the selection screening process that usually relies on hybridoma phage display can be quite tedious and inefficient, just because of the massive number of choices for fragment combinations. If de novo protein sequencing is applied here, it could be used to generate a series of proof of concept antibody fragment candidates, which could determine optimal target combinations. If well-known and functionally validated antibodies against a variety of antigens are de novo sequenced, a panel of recombinant antibody fragments that engage 2 or more targets at once could be generated. Implementing de novo protein sequencing into CAR-T development workflow allows researchers to easily modify and create many CAR-T models by swapping the antigen recognition domain for variable region sequences found in monoclonal antibodies of interest. This also allows for the selection of the best single-chain variable fragment candidates, as well as simple engineering of these regions and detailed optimization of CAR-T models, achieving both enhanced target recognition and reduced non-tumor toxicity. Now, we were the first in the industry to do this using only proteomics. And with our polyclonal sequencing technology, we were actually able to extract monoclonal antibodies from a protein-rich polyclonal mixture for a client that was focused on diagnostic development. Our clients also routinely use our approach in sequencing technology to develop reagents for commercial use, as well as in-house process development. Now, for both diagnostic and reagent development, without a secure stream of validated antibodies, developers risk non-specific binding and batch variability, or even a loss of supply, resulting in wasted time, resources, or worse, project abandonment. Some scientists don't validate their commercial antibodies at all, as they have the preconceived notion that this is an unnecessary step. But we actually met 2 researchers who were a victim to mislabeled antibodies, and skipping this step cost them 2 years and $500,000. Now, in regards to protein characterization, we have noticed more clients have been using us for further information about biophysical attributes. Generally, within the industry, antibodies are characterized based on what they bind to and not their physical properties. So here we see a blueprint of a house and how it can translate to a house that looks quite different than expected. And if we use that analogy in the scientific world, the blueprint is the DNA and the protein would be the unexpected house. More often than not, characterization of target discovery does not include an analysis of the actual physical protein subunit. And as you can see, this leaves the door open to a lot more risk than is ideal. Target discovery should encapsulate all structural information, including protein interactions, as the success and approval of targets is directly related to their ability to reliably and accurately measure specific analytes. Now, the amino acid sequence is globally recognized as a protein's code. There are no ifs, ands, or buts, or variations. The amino acid sequence is the amino acid sequence. So it can be legally used to universally protect newly discovered biologics. And in fact, we have worked with lawyers previously to provide a sequence for patenting. Additionally, our technology is becoming increasingly used to generate anti-drug antibodies, control reagents, and assays testing the clinical efficacy and safety of biological drugs. These antibodies mimic the natural ADA response to the biological drug being tested, and these positive controls typically consist of animal-derived pooled polyclonal antibodies or human monoclonal antibody reference panels against the target protein drug. In cases like this, the specific sequence is critical to the development of assays, and in many cases, regulatory paperwork may require their exact sequence. So I'm going to break down how Rapid Nova's next generation protein sequencing can be integrated within a few steps of the antibody discovery process. So right off the bat, when an animal is immunized through our polyclonal sequencing service, we can obtain monoclonal antibodies targeting the antigen of interest from the blood of the animal. Our technology can be a huge time and cost savings here. With the fusion and propagations of hybridomas, our polyclonal sequencing service can be used to obtain a few high-occurring monoclonal antibodies from a polyclonal mixture, and it can also be used to sequence monoclonal antibodies of interest. In regards to the selection and screening process, obtaining the protein sequences of the antibodies you're more interested in can help you ensure that you're not wasting time. Many scientists use DNA sequencing here, which does work, but as I've discussed, there is room for error as DNA sequencing doesn't pick up most translational modifications or potential cross-reactivity or the presence of noise and extra antibody chains. Now, towards the end of the process, REMAB can be used to provide you with the amino acid sequence of your monoclonal antibodies, if you're interested in recombinantly engineering the antibody, or if your hybridoma cell lines are lost or have died. And our MatchMab service can be used to ensure that recombinantly expressed or manufactured antibodies match the predicted antibody sequence. So to break down what actually happens on our end of next generation sequencing, I'll discuss the workflows and some of the unique technologies that we have that enable us to provide high quality amino acid sequences. So when we receive your sample in our lab, the first thing that we do is determine purity. Now you see the small SDS gel picture there in the left. In a perfect world, when we run an SDS gel, we would see two bands portraying the heavy and light chains of the antibody as seen in the middle column. But often we see what looks like the column to the right, which is that nice big blur that you're seeing. That's the result of contaminants, the like of BSA proteins and BSA antibodies are just noise. Now, in the early days, that would have been a bit difficult for us to overcome. But after completing over 3,000 sequencing projects, we've developed technology that allows us to purify samples like these to maintain our high quality standard. So once we've okayed the purity of the sample, we go ahead and we do multi-enzyme digests. Now, for context, many people that do next generation sequencing will use between 1 to 3 enzymes, while we actually go ahead and use between 5 to 10. And the reason why we do this is because, well, we've worked with a lot of different antibodies, and we found that our little protease cocktail, as we like to call it, really enables us to have a more holistic view of the protein sequence when it's run through our mass spectrometer. We've also developed unique fragmentation techniques with the mass spec that enables us to better determine between variations of amino acids. To determine the order of amino acids, we have proprietary machine learning algorithms, which we like to call novo, that do a first draft of the sequence. And our bioinformatics specialists get involved to ensure that all areas have sufficient coverage and meet our quality standards. Now, for context, 20-fold coverage of the sequence is quite standard in our industry, and that's around 95% confidence. But we aim for 30-fold coverage of the sequence, which is 99 point something percent confidence. In terms of deliverables, we'll lay out the amino acid sequence for you and highlight the CDR regions, as well as any post-translational modifications that we've observed. And what you see on the far right is our peptide viewer, where we detail all the peptide coverage that we've observed and the enzymes that were used. Now, our polyclonal sequencing workflow begins with antigen inoculation. 3 different bleeds are performed, and routine purification is used to extract the IgGs. The antibodies are then affinity purified with antigen-specific purification to enrich for target antibodies. At this point, we use our de novo protein sequencing technology to assess the sample, and it can be used alongside our deep sequencing RNA-seq and NGS data analysis. At the end of that, we're able to deliver full-length antigen-specific IgG sequences, which include heavy and light chain pairing. And we can also go ahead and perform a comparative ELISA in-house and deliver recombinant target antibodies. There are 2 options here. In version 1, we can make use of the combination of genomics and proteomics, and option 2 would rely solely on the proteomics component. We work with clients at different stages of their platforms. So what that really means is that our clients have the capability to do immunizations or synthesis themselves, in which case we are happy to focus on the sequencing side of things. Now, MatchMab is our simple and fast sequence confirmation service. So given a reference sequence, we basically sequence your protein by performing simple sample digests and running it through the mass spec to very easily determine if the sequence matches the expected or not. Now, I've thrown in a decision matrix here, which I'm not going to go into too much detail with, but I wanted to at least put it up for you to see. If this is something that you would like more detail about, or if you'd like me to have a chat with you and walk through it, I'm happy to set up something, so feel free to shoot me a message after the webinar. To conclude with the protein sequence, you can gain direct access to the protein regardless of nucleic material that you may or may not have. It's also used to complement existing antibody discovery pipelines, providing you with additional information about biophysical characteristics, the monoclonalness of your antibody, and the purity of the antibody as well. Protein sequences also enable capturing a more complete picture of the humoral immune response that can really help to inform therapeutics, diagnostics, and reagent development. And finally, last but not least, we can save some animals while we're at it. So that concludes the presentation. Thank you so much for attending. I hope you found it insightful and that you learned a couple of things about DNA sequencing and protein sequencing. My contact details are on the screen, and I'll open up the floor to some questions now. Okay, so we have a question from Dennis. I have about de novo peptide sequencing to the full protein sequence. Do we use automatic assembly algorithms similar to DNA sequencing, or do we assemble the peptide manually half automatically? So it is I would say a combination. There is one part where it's done by our algorithm, but our bioinformatics specialists work with the algorithm to ensure that everything looks the way it should. And if we're not happy with the coverage on the first draft, then we'll go back and potentially run more tests and work with the algorithm again. It's never just the algorithm itself, it's always a combination of the two. No problem. Any other questions? Give you a few more seconds. So we have one question. Who owns the rights to the sequence once we've sequenced someone else's research antibody? Yeah, so we don't hold the rights to the sequence. That information is all yours. We have information on it, but that's about it. So another question, when a post-translational modification is present, does it generally occur 100% of the time in the same position, or are you able to tell the fraction that it is modified? That varies from antibody to antibody. Sometimes we'll see data that says that it shows that there's a modification 100% of the time, and sometimes we'll see data that it shows most of the time. And we can get a more detailed breakdown if that's something that you're interested in.